Okay, welcome back. We just wrapped up Anaphase I. Now let's move forward to the next step here, Telophase I. Now, again, this is going to look familiar to mitosis. In Telophase I, this is the end of the first meiotic cell division. Now remember, meiosis divides twice, and this is the end of the first division. The cytoplasm is going to divide, just like in mitosis. It's going to form two new daughter cells. Each of the newly formed cells has half of the number of the parent cell's chromosomes, but each chromosome is already replicated and ready for the second meiotic division. So once telophase I is done, it essentially is going to shift right into the next step here. And the next step is going to be cytokinesis, the first cytokinesis. This is kind of simultaneous with telophase I. It forms the two daughter cells. If these were plant cells, it would form a cell plate. In animal cells, we have what we call cleavage furrows. And at this point, there's no further replication of the genetic material prior to the second division of meiosis. So no more division until Meiosis II kicks in. And then we kind of do it over again with slight differences. So here are some pictures here, hopefully that help out with meiosis I. We have interphase where the nuclear envelope kind of goes away, the chromosomes condense. We have prophase I where crossing over can happen, homologous chromosomes pair up and exchange segments. We have metaphase I where microtubules attach to these chromosomes here, and a metaphase plate is formed. And anaphase I, where these microtubules start pulling the sister chromatids to each pole. So the homologous chromosomes are going to separate at this point. Okay. Now, meiosis II is going to start next. This is going to separate our sister chromatids. This is similar to mitosis once again, but we're going to see what's going on. There's no interphase 2. You'll notice the only interphase is before meiosis I. So when meiosis I finishes up with prophase I, metaphase I, anaphase I, and telophase I, it's going to go straight into prophase II. There's no interphase 2. It just goes right into prophase II. So here we go, prophase II. At this point, each of the daughter cells are going to form a spindle, and the sister chromatids are going to move towards the equator. So that's just like before. Sister chromatids move towards the equator. The spindles are formed to be used later down the road in anaphase. Okay. Then we have, of course, metaphase II. At this point, the sister chromatids are positioned on a metaphase plate, just like all the other metaphases, in a mitosis-like fashion. So here they are lined up once again. You guys are so smart. I know that you know what's going to happen next. After metaphase II, I'm sure you guessed it, we have anaphase II. And during anaphase II, the centromeres of the sister chromatids finally separate apart. So here's where it's really close to mitosis. These chromatids finally separate, and the chromatids of each pair move toward the opposite poles, and now you have individual chromosomes. So they are no longer paired up. They are no longer diploid. Now we have haploid chromosomes, and that is what we want in meiosis. And I'm sure you have guessed it. We finally have telophase II and the final form of cytokinesis. During telophase II, nuclei form at opposite poles of the cell, and then cytokinesis is going to occur. And once cytokinesis has done its job and finished up, we now have 4 haploid daughter cells. And again, we label haploid with N. You'll probably remember from our last set of notes that diploid would be 2N because it's 2 times the amount of haploid chromosomes that we have. But yeah, now meiosis is wrapped up after you've finished all of those steps. Interphase, PMAT1, PMAT2, and then your cytokinesis finishes it off, turning it into 4 complete haploid daughter cells. So keep that in mind. Don't forget, file this away somewhere in your brain, that meiosis results with 4 haploid daughter cells, 4 complete haploid daughter cells. Now we can look at this picture here. Here's a chart of Meiosis II. Again, at the end of Meiosis I, we have Telophase I, and it starts to split things up. Our 2 haploid cells begin to form. We still have double chromosomes here. During Prophase II, again, spindles form. Metaphase II, the chromosomes line up on a metaphase plate. Anaphase II, the sister chromatids begin to separate and they're pulled apart by the spindle fibers. And at the end of Telophase II and cytokinesis, once again, you have 4 haploid daughter cells. So again, during this round of cell division, the sister chromatids finally separate, creating 4 haploid daughter cells as a result. And each haploid daughter cell contains 1 single chromosome. Okay, now meiosis once again creates very diverse organisms, very diverse offspring, because all those chromosomes are scrambled up and rearranged, which is very important in nature. We want things to be genetically different. We don't want things to be the same. So one way that meiosis creates diverse or unique sex cells is through a process that we call independent assortment. Independent assortment produces 2 to the n power distinct gametes, where n is the number of unique chromosomes. So in humans, n is 23, because if you remember, a haploid cell has 23 chromosomes, a diploid cell has 46, but right now we're looking at haploids. So if there are 23 chromosomes, and to figure this out, we do 2 to the 23rd power, well, that comes out to be 6 million. So there are 6 million unique chromosome arrangements. That is a lot of diversity just from this alone, but that's not where it stops. We are going to look a little bit further here at how this diversity continues. So here's one possibility. We have 2 equally probable arrangements of chromosomes around metaphase I. Around metaphase II, we have several options again, and that's based on what probability we had during metaphase I. If it was like the one on the left where everything's homologous, or the one on the right where it's slightly scrambled up. At the end, your gametes are going to be one of these 4 different combinations. It could be very homologous, they could be very similar, or they could be scrambled up. There are 6 million possibilities, so there's no way to determine what's going to happen. It's just going to be one of those 6 million possibilities. Another way that meiosis creates diverse or unique sex cells is through crossing over. Now, again, crossing over is an exchange of parts of non-sister chromatids. The duplicated chromosomes, they kind of line up really close to each other, and right here, when the 2 chromosomes start to combine a little bit, they get really close, they start getting all snuggled up. We call that a tetrad. And when it's in that tetrad form, the parts kind of swap places. They find the homologous sections of the chromosomes, and they kind of swap places into the other chromosome, where you can see over here a bit, if we look at the red and blue chromosomes, parts of the blue chromosome are now in the red, and parts of the red chromosome are now in the blue. It's because during this crossing over phase, they just start scrambling up portions of their chromosomes. And this also adds to the diversity. So the chromosomes, which already have 6 million possibilities, well, now they start swapping up this way during crossing over, adding even more potential diversity. So the possibilities are virtually endless. All right, this video is about to conclude as well. Make sure you click on the next video to finish your assignment.